Good afternoon. It's, again, very happy to see you coming back to join this wonderful seminar. It's our pleasure to welcome Dr. Jeff Biernaskie as our speaker today. Dr. Jeff Biernaskie received his bachelor degree in neuroscience from the University of Lethbridge in 1998. After that, he moved to Newfoundland and Labrador to pursue his PhD degree at Memorial University. He completed his PhD degree in 2003, studying the effects of rehabilitation on brain reorganization and recovery of sensory motor functions following stroke. It is worth mentioning that this work was the first to describe the critical period in which the brain is most sensitive to rehabilitations and has led to changes in clinical practice following stroke. In the next 6 years after publishing that important work, Dr. Biernaskie underwent his postdoctoral training at the Sick Kids Hospital in Toronto and focused on identifying and characterizing dermal stem cells. Currently, Dr. Jeff Biernaskie is an associate professor at the Faculty of Vet Medicine and Cummings School of Medicine. He is also the chair of Calgary Firefighters Burn Treatment Society. Dr. Biernaskie has a broad research interest, and one of them is in the mesenchymal and neural stem cell biology, particularly the molecular regulation and impact of injury and aging on their function. His research group is currently working on developing drugs to reprogram stem cells within wounds in order to reactivate regenerative programs to improve skin healing. In addition, he and his team are using a large animal model of skin regenerations, which is reindeer antler velvet, to identify factors that enable regeneration and scar prevention. After a busy day of work, Dr. Biernaskie enjoys watching hockey. In fact, he is a die-hard Edmonton Oilers fan. Today,
I knew I'd get that response.
He's very excited to tell us not about hockey, but his research on wound healing using stem cell treatment. The title of his talk is Understanding Mesenchymal Stem or Progenitor Cell Biology in Adult Skin and Their Contributions to Wound Healing. Please.
Thanks very much. Does this have a laser on it?
Yeah, it does.
Oh, perfect. Okay. Thanks very much for the introduction. So that sort of set the wrong tone right at the beginning of my talk. Get the crowd on, against me right off the bat. But yeah, so we do a variety of different things in the lab. We have an interest in tissue repair in a general sense, and the potential role for stem cells in mediating regeneration of various tissues. And so today I'm going to just tell you mostly about the work that we do in the skin and how we might be able to harness stem or progenitor cells to better repair the skin after severe types of skin injury. So I'll tell you about work that we've done to first identify a progenitor cell within the dermis, or the dermal portion of the adult skin. And then I'll tell you about some of the work that we're doing to try to understand how they might be utilized for repairing the skin, but also what their natural response might be to wound healing in the skin, whether they actually do anything when you wound your skin, and if they do, then are there ways that we might be able to activate them or promote their involvement in skin wound healing, and promoting more of a regenerative response rather than our natural response, which is fibrotic scar formation. Okay. So, just on a general sense, the reason that we're so interested in this is because of the impact of severe skin injuries and what they have on a person's quality of life. It's really a devastating thing to have a severe burn or other type of skin injury, and often we think that because they've survived a burn that things are okay, but you don't actually realize the deficiencies and the impairments that go along with that, and the fact that these are lifelong. And as a result of that, you've got immobility, disfigurement, and often underappreciated is chronic itch and pain that's associated with not only the injury but even with skin grafts that are used to treat those wounds. The annual cost of wound healing just in Canada is $4 billion. So this is a huge burden on society to pay for this. Both immediate chronic or the immediate care of wounds, but also the chronic care that's required for people that suffer these types of wounds. And this would include not only an acute wound like a burn, and then the scar that occurs afterwards, but even more chronic wounds like diabetic foot ulcers as well, that require a lot of care and oftentimes eventually result in things like amputation because they're so difficult to treat. So our goal is to really try to understand the diversity of fibroblasts, which I'll tell you a lot about. The whole talk is going to be about fibroblasts in the skin. These are the primary cell type within the dermis. The diversity of these cells within the skin and in other tissues as well, and their dynamic biological functions, and whether we might be able to modulate their natural response to injury in order to improve outcomes from these severe wounds. So really the question is, how can we encourage tissue regeneration and prevent formation of fibrotic scar? And can we exploit a dermal stem or progenitor cell? Does one even exist? So this is what we were thinking about when we first started to do this work. And so I want to just also show you a couple of examples of the wounds that I'm talking about. One is a patient that came into the burn unit over at Foothills Hospital. Okay, so this is a woman, middle age. This is not a burn injury. This is actually necrotizing fasciitis. So she came in with flesh-eating disease on her neck and shoulder. Okay, and to treat that, what you need to do is basically cut out that affected area and repair that with a skin graft, a split-thickness skin graft, which is the top layers of the skin taken from another part of the body and then transplanted over that wound. And essentially that provides a covering over the wound so that you can prevent infection. But the function of that skin, as you can see, is really, really poor. Okay, so you can see the amount of contracture that's within the graft. So this is actually 7 surgeries, 7 different grafting surgeries to actually repair this wound. And this is the outcome that you get. This is horrible. You can see the contracture on her mouth, so she has a lot of difficulty eating and talking just because it's pulling down on her mouth. Also her shoulder is pulled up from the contraction, so she has immobility in her arm, preventing her from going back to work. And then her breast as well is also pulled up, so the aesthetics of that is also displeasing to her. So there's this is a real disability. This doesn't only affect humans, and because one of my appointments is in the vet school, I'm obligated to tell you this also affects animals. This is a performance racehorse that's had a distal limb injury. And so you can see here what happens is that they produce too much fibrotic tissue, or they have a very robust response, wound healing response, in that the fibroblasts tend to proliferate too much and they don't allow this to actually reepithelialize. And what you're left with is what's called proud flesh, which is this sort of bulbous chronic wound that's really difficult to treat. And so in both cases you're left with chronic wounds, and in this case this animal is likely finished in terms of being a racehorse. This is a huge cost to the consumer, or to the owner, and likely this means that this animal is going to be euthanized. So we want to try to figure out ways for both humans and animals where we might be able to enhance repair, and we can do this in a variety of different ways. So does a stem cell actually exist in the skin, or a dermal stem cell I should say, because there's been decades of work been done on epithelial stem cells. And so this is just a schematic showing a cross-section through the skin for anyone that isn't familiar with the skin. So this is the epidermis, the outer layers of the skin here, and then beneath that is the dermis. Okay, and then beneath that is what's called the hypodermis. It contains a lot of fat and other fibroblasts. Okay. Embedded in the skin is hair follicles, glands, many nerve endings, and blood vessels. Okay. And so this is really a complex tissue and a very important sensory organ for our bodies. But But what's unique is, one of the unique things about the mammalian skin, and this is all mammals, is that they have these hair follicles that undergo continuous regeneration. So this is one of the only structures in the adult body that continually regenerates. So it undergoes degeneration and then regeneration. And I'll show you a schematic of that in a second. But the reason that's important is because in order to do that, this structure has to harbor a number of different stem cell populations within it. Okay? And so the epithelial stem cells that would reside in this region here, they basically give rise to the entire structure of the hair follicle. So there's periods of growth where the follicle will actually start to, those epithelial cells will start to grow, and they basically build the hair follicle in a downward fashion. There's also, actually I'll just move to the next slide to show you. So this is the schematic of what this regenerative cycle actually looks like. So this is the resting hair follicle. This is where it's sort of dormant. And then the epithelial cells are activated by signals that are unknown by dermal or mesenchymal cells that sit in these blue and red cells here. So this is called the dermal papilla, and then the red cells around here are called the dermal sheath. Okay? So these cells send signals to the epithelial cells that sit here, and then they start to build the hair follicle in a downward fashion until they reach this mature hair follicle. Okay? Then what's really interesting is that this whole bottom half of this follicle undergoes complete degeneration. So almost all of those cells die, with a few exceptions being these mesenchymal cells that stick around. There's a subset of mesenchymal cells that stick around that I'm going to tell you a lot about in the next few slides. And then these epithelial stem cells. And then 2 weeks later, or several weeks later, this whole process starts again, and it continuously regenerates. And so what's happening is, as these epithelial cells are building this follicle, they're migrating downwards, and they basically take a U-turn, differentiate, and then they build the hair fiber that then just gets pushed out of the hair canal. Okay? So this is how you're continually building and growing new hair. So we wondered, if there's anywhere in the skin that might harbor a dermal stem cell or mesenchymal stem cell, it must be in the hair follicle because of its regenerative capacity. And really, this hadn't been looked at before. But these red cells that I've sort of drawn out here, in order to build this follicle, they need to increase in numbers by a huge amount. And so they must be proliferating. There must be some cell type that's able to proliferate as the follicle is regenerating in order to populate all of these red cells as the follicle actually builds itself. And the same with this blue structure here, this dermal papilla, which is really important for sending the inductive signals to tell it to regenerate. That population also expands in number, and yet there's never any sign of proliferation there. So there must be some. We hypothesize that there must be a stem cell outside in this red structure here that's actually functioning as a stem cell. So we did a series of experiments using different transgenic approaches to actually fate map the cells in that dermal sheath, that fibroblast layer that wraps around the hair follicle, to ask, is there a stem cell that resides in that structure, and are they important for regeneration of the hair follicle? And so this is an alpha smooth muscle actin promoter that essentially drives Cre recombinase that will activate a yellow fluorescent protein. Okay, so any cell that expresses this gene, alpha smooth muscle actin, will be labeled green when we give a drug called tamoxifen. Okay, so we inject the animals with tamoxifen, and what you can see is that after tamoxifen, all of these cells that wrap around the hair follicles turn green. Okay, so now we can follow these cells and their behavior over long periods of time to see what they actually do. This is just a close-up of some of these cells wrapping around the outside of the follicle. So what we did is look at sort of a time course of these cells. And so this is at the very beginning of one of these regenerative cycles. We label the cells. Then we look a few days later at what those follicles look like, and you can see that those few cells that are labeled initially start to divide and start to expand. And actually they will layer around the entire length of the hair follicle. Okay? And you see it probably a bit clearer here. Then the follicle undergoes degeneration, like I told you about. So most of the cells in the bottom half degenerate. It basically collapses like a sock. And then a lot of these, many of these green cells that we initially labeled also die off. But there's a small subset that sticks around. So this is now back to this resting telogen follicle. There's a couple of these cells that hang around here. Now what's really interesting is what happens in the next cycle when you actually follow what these cells do then. So at the beginning of the next cycle, it's only those green cells, those cells that have been retained in the hair follicle, that actually start to divide again. Okay? So this is where we give a drug called EDU, which is the thymidine analog. So any cell that starts to undergo mitosis takes up that drug. Okay? And it's only those green cells that are taking it up and starting to divide again. But when we then look at the follicles that are generated in that next cycle, we see that those very few cells that were retained, that were labeled with green, are the ones that are repopulating the entire dermal compartment of the hair follicle. Okay, so all of that dermal sheath around the follicle, and many of the cells within the dermal papilla are being regenerated by those few cells, those few green cells in the beginning. Okay. We could then do a couple of other experiments where we did some genetic tricks where we just looked at individual stem cells within the hair follicles to find out whether these cells could actually self-renew and whether they were multipotent, giving rise to multiple cell types. And I won't get into the details, but what we can see is an individual cell is actually able to give rise to multiple cell types. So here's an example of where one cell that we labeled gives rise to a variety of new stem cells. So this is where the stem cells actually reside, is at the bottom of the follicle. In other cases, they differentiate and they give rise to these dermal papilla cells. Here they give rise to dermal stem cells, but also the cells up in the dermal sheath, so they're making progeny that are migrating away. And then here, all of them make dermal sheath. And then in other cases, they don't divide at all. They just sit there and remain quiescent the whole time. So they do a variety of different things. But what these experiments told us is that there's a self-renewing dermal cell in the hair follicles that's able to give rise to the majority of cells within the hair follicle and enables regeneration of the mesenchymal components of the hair follicle. The other interesting thing is that you can actually take these cells out and put them in a dish, and when you grow them in the presence of growth factors like fibroblast growth factor, they'll actually divide, and they stick to one another, and so a single cell will divide multiple times and give rise to these spherical colonies of cells. And so these are just called, we've called them skin-derived precursors in the past, but it's really dermal progenitors that have divided and given rise to these colonies. Then you can break down each of these colonies into single cells, and they'll generate new colonies. So they're able to self-renew in vitro as well. And so this got us thinking about maybe these cells could actually be taken out of the skin and actually used as a source of new dermal cells that could be transplanted into skin wounds to actually regenerate new dermis. And so one of the things that we did was to ask, well, do they actually retain their function after they've been cultured for long periods of time? So what we did was to grow these cells in vitro from mice that were expressing a green fluorescent protein. Okay, so all the cells are labeled green. And then if we take those cells and then we combine them together with epithelial cells, so the cells from within the epidermis, mix them together, and then transplant them into an immune deficient mouse so that they don't get rejected, what we see is that those dermal progenitors are actually able to instruct the epithelial cells to build a new hair follicle. So this is a graft of these dermal progenitors together with the epithelial cells that actually formed hundreds of new hair follicles that were all single cells, but are able to provide the appropriate instructions to actually rebuild hundreds of new hair follicles. And so this is just a close-up here. You can see that all of the dermal components of the follicles are actually regenerated by these cells. So this says that even after long periods of culture, that these cells can actually retain their functionality. Okay. So can we actually put these into wounds to actually repair the skin? Or alternatively, could we actually stimulate these resident cells in the skin to actually be recruited into wounds and actually improve our natural response to wound healing? Do they actually participate in wound healing at all under normal circumstances? These are questions that we were wondering. So the first thing we did was to ask whether we could generate these same cells or isolate these cells and expand them in culture from human skin, adult human skin. And so we developed conditions that we could do that. So this is growing them in a couple of different growth factors, one being platelet-derived growth factor, another being fibroblast growth factor. But you can take skin from almost any age. So we've done it from patients as low as 18 years of age all the way out to over 80 years of age. We take a biopsy of skin, isolate the cells to single cells, and then grow them in culture, and they'll form these same colonies that we see from rodent skin. And then they can be passaged multiple times. So you can generate millions and millions of these cells from a very small starting population. And when we look at all of the proteins that they express within these colonies, they look very similar to what we see in rodents, and they're very classic sort of fibroblast type markers. But they also express some markers that differentiate them slightly, and that's markers of self-renewal or a marker that's sometimes associated with other stem cells found in the body, which is SOX2. So they have a lot of similarities to other somatic stem cells, but they are truly a subtype of fibroblast. So what we wondered was, I told you about that initial, that woman that came in and had all of those skin graft surgeries, and so what we wondered was whether we could use these cells to actually make that procedure better. Because what happens is you take a split thickness skin graft is really, it's almost like a motorized cheese slicer that you use on somebody's leg or on their back or on their buttocks to take a piece of skin that then is sutured on top of the wound. And so when you do that, you're taking only the epithelial, only the epidermis, and very little of the dermis. And so the dermis provides so many nutrients and support structure for the epidermis that when you put that graft on there, that's why it's so dysfunctional, is because it's missing that mechanical and nutrient support that's provided by the dermis. And in the absence of the dermis, you just get this filling in of scar tissue, which is why it contracts so badly. And so what we wanted to know was whether we could populate the graft with dermal progenitor cells and then transplant that and see if the graft function was improved. Okay, so we developed a mouse model where we took human skin graft from the operating room at Foothills Hospital. Take what would normally be put onto an actual patient, took the remnants of what was left over, and then put that onto mice, so immune deficient mice. So this is an example of one of these skin grafts, human skin graft put onto the back of a mouse. This is 1 week, so you can see the suture still there, 4 weeks and 8 weeks. See the cells that we're transplanting for periods of time after the graft. And then put those onto a scaffold so that it gives them some structure, put that into the wound, and then a split thickness skin graft goes on top of that, and then we look at the fate of the graft over a period of time. So this is two examples of two different grafts that we've done. So the green indicates the epidermis. This is the outer layer of the skin. And then the red is all of the transplanted cells that we put in. So these are adult human dermal progenitors that were transplanted into the graft. In this case, we just put the cells in by themselves without a scaffold. And you can see that there's very sporadic retention of cells within the graft itself. But if we put them on a scaffold, so this is just a collagen scaffold that gives them some sort of structure to actually adhere to, you can see that it actually fills in the entire depth of the dermis. So the cells actually go in there, they start proliferating, they migrate, and they fill in the entire depth of the graft. And so they're essentially making a neodermis that fills in the skin graft. And so now what we're working on is actually trying to understand whether these are actually functionally better than a skin graft by itself. And so we're working with people like Elena DiMartino's group in bioengineering to actually do sort of viscoelastic property testing on the graft. So do they actually retain are they as flexible with cells, or are they more flexible when you transplant cells, or actually are they worse? We want to know about all of these different sort of structural properties to know whether that graft might be better functioning with cells or without cells. The other thing that we looked at was in terms of this, this itch and this pain. And so we analyzed animals over a period of 3 months after they'd had either a graft with cells or without cells. And what we did was something similar. We just put them into a box and then just filmed them for 20 minutes at a time, and then looked at how many times they went and scratched their graft. And when you actually transplant cells into the grafts, and actually what's interesting is it doesn't have to be the dermal progenitor cell, it could be other fibroblasts as well. But the presence of a transplanted cell within the graft actually reduces itching by about half. So you see these guys just vigorously going after their graft, trying to itch this thing, and sometimes they'll actually destroy the graft because the itch is so bad, and it may actually there may be a pain component to it as well. But what was remarkable is that we could actually reduce the bouts of itching by about half when we transplanted cells. So we're trying to follow that up now, primarily to see if we can get better integration of these cells within the grafts, so that we can get a more reliable outcome and see if we can improve not only the mechanical properties of the graft, but also the pain and things that I mentioned. So the other aspect that I want to tell you about is the alternative way of thinking about this, not necessarily transplanting cells in, but can we actually activate cells to naturally respond to a wound? So are the cells within the hair follicles responsive to an injury? That was the first question. Do they actually, if you make a severe wound, do they actually migrate out of hair follicles and participate in wound healing? And then our ultimate goal is, if they do, can we actually enhance their response? Are they actually regenerative in any way, or are they just contributing to more scar? So this is essentially the experiment we did. This is using exactly the same alpha-synuclein mouse that I told you about before, where we could label those cells, and then what we did was label them with the tamoxifen application. 4 weeks later, we made a large skin wound. Okay, and then we followed the healing process for between 4 and 12 weeks after that. So this is a schematic of the wounds, and this is an actual image of what these wounds look like. So what you're going to notice is this is a really large wound. This is a 2 centimeter squared wound on the back of a mouse. There's a reason. But what's cool is that in the center of the wound, they actually undergo true regeneration. So, and this is, I think rabbits do this and mice do this. But what happens is that in this central region, you actually can regenerate new hair follicles, you can generate fat, and you get actual reinnervation of that region as well. And so this is, potentially a model of how we might provide some hints as to how we might regenerate skin on a larger scale, and how we might move this towards application in humans. So if we can understand what's different about this phenomenon than, say, this phenomenon, then we might have some idea of how we might promote regeneration and prevent scar. So we made these very large wounds in these mice, and then looked at the response. So this is the normal wound, or the normal skin following tamoxifen application, which we've seen before. And this is what it looks like within the wound. So the white dashed lines indicate the edges of the wound, okay, here and here. The red is the epidermis. But what you'll see is that green cells from out here have actually migrated into the wound, okay? So there's not a huge number of them, but there are still fairly reasonable numbers of cells that have migrated from the follicles actually into the wounds themselves. So we looked at a 4-week time point, or actually, sorry, this is at 1 week after. What's interesting is that what we had hoped would happen is actually happening, in that if you look at the hair follicles that are right next to the edge of the wound, you see that the green cells in those follicles, relative to the follicles that are further away from the wound, are proliferating like crazy. So this is a higher magnification of this follicle here beside the wound. You see that many of these green cells are actually peeling off of the follicles, and they're actually staining with Ki67, which is a proliferative marker. Okay, so the cells are migrating off of the follicles and actually towards the wound. So there's something that's attracting them into the wounds. And so it looks like they're actively being recruited to repair the wound. So then we looked at a 4-week time point at these wounds. So this is a cross-section of the back skin. This is the wound itself. So here's the two margins. And this is the central region that I talked about that's actually undergoing regeneration. So what you'll see is these little structures here. These are actually newly formed hair follicles that have formed in the wound itself. So this is a bit closer image here. So there's about 7 follicles that have formed there. And this is a higher mag of these follicles here. So you can see those green cells that have originated from those hair follicles outside the wound are actually moving in and participating in this regenerative process and forming these hair follicles. Okay. And without these guys, without these specialized mesenchymal cells, the follicles won't regenerate. So what this means is that cells are moving into the wound and actually retaining or acquiring regenerative properties that they didn't have before. Okay, so they're moving in and then providing the appropriate signals to the epithelial cells to basically undergo morphogenesis again, or recreate skin embryonic development. Essentially, that's what's happening here within the wound space. But what you'll notice here is that it's not really what we had hoped to happen. So if you look at, you can see this whole structure here, the dermal papilla, and maybe even better in this example here. There's a number of cells that aren't green, and all of the cells that surround the hair follicle, the dermal sheath, that we would expect to be green are not. So what this tells us is that, in fact, even though these dermal progenitor cells from the follicles are participating in this process, they're really playing a very minor role in this whole thing. So this is less than 20% of the cells within the wounds, and certainly less than 20% of the cells that are actually participating in regeneration. So these guys are playing a very minor role. So this tells us that there's actually another population, dermal progenitor population, in the skin that's really the primary player in wound healing and the primary player in regeneration. And what's really interesting is that this cell type is likely never associated with a hair follicle before, because we've labeled all of the hair follicle dermal cells, and yet we're seeing other cells that are actually recapitulating or regenerating this hair follicle. So we went back to some of our other studies to ask, where are the rest of the cells coming from? And is there another progenitor population in the skin that we need to be focusing on? Because it looks like the hair follicles are really not the primary source of wound healing fibroblasts. And when we did a series of gene expression analyses on the dermal stem cell population. So we isolated these cells and we did RNA sequencing and looked at what cells were, or what genes were really enriched in this stem cell population. What we found was this gene called HIC1, or hypermethylated in cancer. This is a tumor suppressor gene that's often methylated in various types of cancer. So what it does is it puts the brakes on proliferation. And when it's mutated, the cells proliferate out of control, which is why you get a cancer. But here it probably plays a role in regulating a stem cell, in that it allows it to proliferate at some times, and then it maintains it in a quiescent state in other times, which you can imagine is important for regeneration of the follicle, where it needs to be active at some times and not active at other times. And so we found that this gene HIC1 was really highly expressed in this dermal stem cell population. And so we looked at, we generated these HIC1 Cree mice, which allows us to look at where HIC1 is actually expressed in the skin. And not surprisingly, we see that the same place where our dermal stem cells are, we see labeling for HIC1, and all of the cells that come from those dermal stem cells are also labeled with this HIC1 crete. This is again in just one of these resting follicles. You can see labeled cells here where the stem cells reside. But if we look at the skin on a whole, what we see is that not only do they label the progenitor cells in the follicle, but they also label a population of progenitors that sit in the very deep layers of the dermis. Okay. And so if we look at if we just give a pulse of tamoxifen to look at where it's expressed, we see a very small number of cells scattered throughout the deep layer of the dermis that are labeled. And these cells undergo slow proliferation, and what they do is they give rise to new fibroblasts and new fat cells. Okay, because when the skin is regenerating follicles, it gets thicker and thinner, right, over these cycles, and so you've got to populate it with new fibroblasts and with new fat, and these are the cells that are actually doing that. So what we asked was, okay, if it's not the fibroblasts in the follicles that are playing a role in wound healing, maybe it's these cells down here. And so we did the same wound healing experiments using this mouse now, and then asked, how do these red HIC1 positive cells or HIC1 expressing cells participate, and what do they contribute to regeneration? And what was really remarkable is that the majority of cells within these wounds are coming from this HIC1 expressing progenitor population. Okay, so here's a couple of examples of wounds here. You can see that the bulk of cells within the wound are actually coming from this HIC1 expressing population. This is just a close-up here. You can see the epidermis. So they don't make any epidermal cells. They only make the dermal fibroblasts that are repairing the dermis. They also make some of the vascular cells within the new dermis. But when we look at this regenerative zone, so we go back to this large wound and we look at this regenerative zone here, we look at those follicles, we see that all of the cells, all of the mesenchymal cells within these newly generated follicles are actually coming from this HIC1 positive population. So this is interesting for two reasons. So this is just these cells here, and then the cells surrounding the follicles as well. So this is interesting because the dogma in the field is that fibroblasts in the lower part of the skin are all fibrotic, that their natural response to injury is to contribute fibrotic scar, because they're very hastily recruited into wounds, but also they have a real propensity to make extracellular matrix. So they move in there, and then they just start cranking out collagens and other extracellular matrix, and they form this really robust scar. So they've always been thought of as being the source of fibrotic scar. But what our data is telling us is that, in fact, these cells are not biased to making scar, but it's the cues that they encounter when they move into the wound that actually tells them to become either regenerative or fibrotic. Because if you look at the outsides of the wounds in those fibrotic zones, they don't induce hair follicles, they don't make that inductive mesenchyme, but they just make fibrotic scar. But when you look at the regenerative zone that has these permissive signals, they're actually able to engage in a regenerative response, and then induce or instruct these epithelial cells to regenerate follicles. If you look later on, in the wounds, these same cells also start to make fat, which is something that normally doesn't happen in small wounds, and certainly not in human wounds as well. So these cells are not only able to generate the inductive mesenchymal cells, these regenerative mesenchymal cells, but also are able to give rise to new fat. And fat is really important in the skin. It provides a huge number of hormonal regulatory signals to other cells within the skin. And so if you want to recreate a functional piece of skin, you have to repopulate the fat within the skin. The other interesting thing was that how this is happening. So the question was, how are these cells actually becoming regenerative again? They've never actually been associated with a hair follicle, and yet they move into a wound and they somehow acquire this ability to become regenerative, and they acquire this mesenchymal fate while retaining also the ability to make fat. So how do they do this? And so one of the hints that we got was when we just started to look at genes or proteins that are only expressed in the embryonic skin. So this one being SOX18, this is so this is embryonic skin. This is the very beginning of a hair follicle that's being generated. And you can see that those specialized mesenchymal cells express SOX18 at this stage. But if you look at any other time point after birth, you will never see SOX18 re-expressed again. But in this case, when you've got these cells moving into the wounds, only in the regenerative zone, they start to reactivate SOX18 again. That's here. So this time it's shown in white. But all of those cells within that, the mesenchyme, are now activating genes that are really only expressed in embryonic development. So what looks like happening, what's happening is that those cells are being recruited into wounds. In one environment, they see fibrotic signals, but in another environment, they're actually enabling those cells to reactivate gene expression programs that only occur during development. And then as a result of that, they're able to recapitulate skin development and form new follicles, new glands, as well as new fat. And so we went a little bit further. This is just some data that we're working on now, which is trying to understand how this is happening. So at the genetic level or the transcriptional level, how are these cells actually acquiring this regenerative capacity or competence? And why are they not becoming fibrotic? And so we just went in and took cells out from either these fibrotic regions or from the regenerative zones. And we took them at a time before any of the hair follicles are being formed and asked, okay, when they're sort of primed to stimulate regeneration or to become fibrotic, what are the gene expression programs that are being activated at those times? And so this is just, so we did this by using what's called single cell RNA sequencing. And so we took thousands of cells and then looked at gene expression in every one of those cells. And so all of the cells are sort of plotted here. And this is a little bit strange to look at. I'm not going to go into all the details, but what it allows us to do is to actually cluster out these different populations of cells from these different regions, and then from uninjured skin or from other types of wounds that are essentially only fibrotic. And so when we look at cells that are from within this regenerative zone, they're shown here in green. This is the cluster here. So their gene expression program is entirely different from the cells in this fibrotic zone, which are in the blue area here. So you can see that on the transcriptional level, they're entirely different. And then the purple is uninjured skin. So you've got, what this is sort of summarizing is different gene expression programs that are being activated. So here, this is the normal fibrotic response shown in all of these cells here. This is, again, like thousands of cells. And then this is the cells that are undergoing this regenerative process. And so we can look at each of those clusters separately and then start to ask, okay, what are the genes that make up these different populations? So this is an example of some of these. So this is genes that are essentially involved in maintaining homeostasis, or sort of a homeostatic state in uninjured skin. Here's another set that's involved in the fibrotic response. And then here's another set that's involved specifically in those cells that have acquired this regenerative state. What's interesting is that almost all of these are activated during embryogenesis as well. So if you look at this whole signature, and there's about 60 genes that are really unique to this regenerative profile, more than half of them are genes that are active very early on in embryogenesis. So we think that that's what's happening, is that you're getting this unmasking or this enabling of embryonic gene expression programs that allow them to do this, to become regenerative. And you can see that when you look at individual genes within this regenerative cluster, so like I said, regenerative cells are here. If you look at some of these genes, you can see that you can pick out specific genes that map specifically to those regenerative cells, and then other ones that map specifically to the regenerative cells. So one being IGF2, critical for these regenerative cells. PLEC3B, again, these are important for at least part of the regenerative cells, but also important for are also activated in just injury itself. Retinoic acid signaling seems to be really important in this process, actually. So we see a huge array of genes that are activated within the retinoic acid network. And then again, IGF signaling is also very, very active, specific to the regenerative population and not in the scar-forming populations. We did this slightly differently, looking at a transcription factor network, where we look at all the downstream players, so hundreds of different transcription factors that are related to, that are downstream of individual transcription factors. And what we see is another one that's interesting is the SOX810 and HOX89 are what we think are really key for establishing this embryonic capacity for regeneration. So this is sort of the early stages. These guys are activated early on. If they're activated, they enable these cells to be regenerative. And then later on, they start to initiate more specialized transcriptional programs that allow them to become inducted. So this is what enables them to start telling epithelial cells to be regenerative and to rebuild these structures. So things like the RUNX family of transcription factors are really important for that. So the reason that we're so interested is that what we want to do, and I'm going to end right here, all of our efforts now are really trying to figure out or trying to identify drugs now that can specifically activate these same regenerative programs. So we know all of the different transcription factors that are active and some of the downstream targets of those genes. And so now if we can find drugs that we could actually apply to those wounds, maybe we could persuade those fibroblasts from becoming fibrotic and push them to becoming only regenerative. So, and expanding that regenerative zone from being just that central region to including the entirety of the wound. And vice versa, also just applying drugs that might inhibit or block the genes that seem to be associated with the fibrotic phenotype as well. So just trying to modulate those two states. So what I've told you is that there's a multipotent stem or progenitor cell that's in the dermal sheath of hair follicles that's critical for regenerating hair follicles, and is able to repopulate the mesenchyme of the hair follicle. We think that, or we can see that these cells are actually activated after injury, but they really play a minor role. And this is actually, this wasn't necessarily unexpected, but is actually a good thing for us, because in humans we don't have the same density of hair follicles as mice do. So this might, if they had been a real active player, this might have been beneficial for mice, but in humans this probably isn't a feasible source of cells to sort of endogenously activate to repair wounds, because I don't think we have enough of them to actually make a significant improvement. But the fact that we've got these progenitors that are within the sort of deep layers of the dermis, and there seems to be a large number of them that are associated with the vasculature, they, I think, are a reasonable target to actually go after to promote regeneration. And then I told you that we've got this, like I said, this HIC1 positive progenitor population that's in the deeper part of the skin, that's associated with blood vessels that generates fat and fibroblasts. We think that these cells harbor a sort of a latent regenerative capacity, and that these are going to be really important for promoting dermal regeneration. Again, the fate of these cells in wound healing, we think, is really driven by factors that are within the wound environment itself. And so one of the things we're doing now is to try to dissect wounds to figure out what are those secreted factors or soluble factors within the central regions of the wound versus the outer parts of the wound, and how might those be driving these different fates, as well as some of the other cellular players that are there. So we think that one of the differences is that you have a different composition of immune cells in the central regions versus the outer regions of the wounds, and that's what's really driving these cells to become fibrotic or regenerative. And so we're now looking at trying to develop drugs that we could potentially use to promote a regenerative phenotype. So I'm happy to take questions, and I just want to acknowledge the people that have done all this work. It's really Sepede Abassi and Andrew Hagner, two PhD students in my lab, along with Prajay Shah and Joe Stratton, who have done all the bioinformatics and a lot of the molecular biology on these projects. So I'm happy to take questions. Thanks very much for having me.
Floor is open for question.
Might be a naive question, but logistically, how do you harvest the stem cells from the base of your health? What is that process like?
So you'll take out the entire piece of skin. So we can do it from a biopsy as small as 8 millimeters, but we haven't gone smaller than that. But so if you take a biopsy, and we'll also use larger pieces of skin, mince up the skin to small pieces that will include the entire depth. Actually, first, before that, we use an enzymatic treatment to remove the epidermis. We don't want any of the epithelial cells, so we just basically throw those away. And then you use like a collagenase disphase enzyme treatment to dissociate or digest a lot of the extracellular matrix and liberate all the cells. And essentially, they'll just peel off of the follicles with that enzyme. And then you resuspend them in a like a buffer solution, and then count them to a certain density, put them in media, and the cells grow really well in certain factors. Yeah. So yeah, it's actually not that difficult to actually grow them out. I mean, I say that it's been like 8 years of work to actually define these culture conditions and stuff, but they grow pretty reliably from both human and from rodent. And like I said, we can get them from various ages. It declines a lot with age. So one of the things we're interested in the lab is the impact of age on the function of these cells, because as you know, you lose your hair as you get older. At least most men do. And one of the reasons is because these cells become dysfunctional. So there's a lot of interest from like cosmetic companies and things like that about how you might maintain the functionality of these cells so that you don't lose your hair, or could you potentially reinvigorate them in order to repopulate the follicles and reestablish growth. But also with wound healing as well too, because we think that they actually, all of these cells, whether they're from the follicle or from the deeper parts of the dermis, are reduced greatly in number with age. And so ways of maintaining those numbers so that you can maintain your ability to heal wounds would be really important. So that's another thing that we're sort of looking at. Yeah, I do. Yeah, that's a good question. That's something that we've thought a lot about, particularly. So as they move up the follicle, they're sort of stretched around it, and we think that a lot of the sort of mechanical properties of the follicle at different regions causes them to differentiate. So as soon as they have that sort of tension, then they differentiate and they commit to they're no longer a progenitor stem or progenitor cell. We think that's one of the triggers. And so as the epithelioles are driving downwards and they're building that follicle, it's that the change in mechanical property is what's driving the other ones to differentiate. And then at the bottom, they have a very unique sort of extracellular matrix environment that they create there that's actually very soft, and I think that's what's allowing them to stay as a stem cell. And so we've done a couple of things looking at growing these cells in either soft or stiff substrates. This is work we've been doing with Mike Callis' group, and if you put them into a soft gel versus a more stiffer gel, you get immediate differentiation when they're on a stiff substrate. And then on the soft substrate, they grow like crazy and they maintain their undifferentiated state. So, and you know what? I think actually what's more interesting is the mechanical properties within the wound itself. And so the tensile strength when they hit it, or the stiffness of the substrate in the center of the wound may be very different than it is on the outer parts. And so, we'd love to look at some of those things. That's sort of beyond our level of expertise, but we'd love to see how changing the properties within the wound might change their response and maybe expanding the regenerative ability. Good question. Absolutely. We started to do some of that too, because these, like the commercial, like this is a huge market. If anyone here is a bioengineer that can develop, some unique, tailorable scaffolds. We had a postdoc working on it for a while back. There's a huge industry there because it's basically just collagen scaffolds that, bovine collagen, they're really not that effective. In fact, the one that we use, which is an FDA-approved scaffold, the cells don't like it. They don't survive very well on it, and when you put them into patients, they form scar really well. So they're not really if you could find something that wasn't so scar promoting, then it would be useful. So I think that's a really, there's a lot of research into that in the field.
So you said you find these stem cells in the wound itself and also the hair follicles that start to grow in there. How come usually when you have a scar, there's no hair on it?
So this only happens in this model of mice. So when you make a large wound in a mouse, which is why we're studying this thing, because normally in a mouse, you make a small wound, it forms a scar just like it does in humans. But for some reason, the large wound in the mouse, there's a different process that happens within this region. So we just use this as a model to understand what those cues are, what those signals are that allows us to be regenerative. So whether this is truly applicable to the human situation, I think the things that we learn from here, we're not going to necessarily start regenerating all of the follicles and glands on a person. At least that's not our goal. Our more reasonable goal would be simply trying to understand or recapitulate these processes that we see in the mouse in order to improve the outcome in terms of lessening the severity of the scar, so that you could get maybe better mobility. I think it's probably a long shot that we're going to start regenerating all of returning to the same density of hair follicles. But I think even if we can just minimize the severity of the scar and enable better reinnervation of the grafts, that we could probably greatly improve outcomes and quality of life for patients. So we still have a fair amount of work to do yet, and we're just sort of being able to translate some of this.
Did I understand correctly that at least in culture, these cells are somewhat resistant to senescence?
There may be a subset that undergoes senescence. Certainly in vivo, they undergo senescence, which is what we when these are subset are undergoing senescence and then death with advanced age. But what happens in vitro is not that they senesce, but that they get transformed. And so they acquire mutations that allows them, and we're sort of selecting for that, right? So there may be ongoing senescence as well, but because we grow them in, with growth factors that are driving their constant proliferation, we've got cells that are dying, but we're just throwing those away, and we're selecting for cells that are able to grow really well. And so, I think the bigger concern is not necessarily senescence when we're trying to grow these cells for transplantation, but making sure that we're not selecting for cells that have acquired an oncogenic mutation and are now tumorigenic, and when we transplant them, they form something that they're not supposed to.
I was just wondering, because it seems like it could be another cell source for very interesting to see other cells that really don't necessarily senesce and can be attractive, although it sounds like there's not a ton of them.
But they do proliferate a fair bit, so you can generate large numbers of them. What's interesting is, even for, I don't know if you're familiar with induced pluripotent stem cells and things, right? So
Now these cells, because they express they endogenously express things like SOX2, which is one of these reprogramming factors, they're very easily reprogrammed to a pluripotent state. In fact, I think that they only require a single factor, OCT4, plus some media components in different drugs. And so they may be a reasonable source to actually make the reprogramming process a bit more efficient. So yeah, they may have other uses as well too.
Yeah, because senescence is a big issue with diagnoses.
Yeah.
From what I understand.
What's the vascular shape of light in the center edge that generates relative to the edge that has the issues?
Yeah, we haven't looked at it sort of systematically. I mean, it's very well vascularized, but the whole graph, or the whole wound gets pretty well vascularized. I don't know if if it has a higher density. We haven't really looked at it because what we're trying to do is to develop ways of being able to. What we want to do is clarify the whole wound with a tissue clearing process, and then be able to map all the vessels in sort of 3 dimensions, so we can actually look at what's happening in these different regions. We just haven't done that yet. So I can't really say whether there's a difference in vascularity between the 2 regions.
So there's some like dense stem cell lesions and vascular, it's also the oxygen
Yeah.
differentiation first occur. Proliferation
Oh yeah, these are very well vascularized. The entire depth of the wound. There's not. I mean, at first glance, it doesn't look like there's a difference between those regions in terms of the density of vessels. But you're right. Like all of those one positive cells that I told you, the bulk of those cells are sitting on vessels, and whether they're some kind of a pericyte or something like that, I think it depends how you define a pericyte. But I think. I mean, the other people that are working, our collaborators that are working on these cells, think that they're actually sort of upstream of the pericyte, that these cells are a progenitor that's actually giving rise to pericytes along the vessels, and that they're also able to do these other things. But it still needs to be refined.
Can I ask a question, please? I have one question. So to follow up Sabrina's questions, do you have the figure of the mouse after the wound injury to see how the scar formed, the big wound that you could create? And the other one is,
So say that again?
do you have the pictures, like you take a follow up how the wound regenerates or heal up by itself, and after the end of it, do you see like how the wound is healed? Like, is there a scar only in the periphery?
Yeah.
And you have hairs in the middle, or how does it look like?
Yeah, so it's exactly that. You've got basically, it's like, it's bald in the periphery, and then you've got this central region, and you've got, between 20 and 30 hair follicles that are hairs that are pushing up through the.
That's interesting because I injured myself before. I fell from a bike, and then my skin was just completely removed from my bike. And then after that area, in the middle, the entire area is without any hair.
Yeah.
So why is it?
Yeah.
So why is it different between human and mouse?
Yeah, with a full thickness injury in human, you won't get any hair. Once those follicles are gone, they're gone. Yeah.
So the regeneration capability is completely different between mouse and human in this sense. It's
Yeah. Yeah. So, yeah.
okay. Just nothing. wait.
Can I follow up on
Yeah,
that?
sure.
Because, cyclists wax their hair, presumably that when they do fall, that skin healing is much better compared to when you leave your hair on. So is there any scientific evidence that is actually correct? I mean, that's one of the reasons why all the cyclists can have, of course, beautiful muscular legs, shave their legs, and then they look even more beautiful. But the idea
I thought that was for to be more aerodynamic and not have this drag of all the hair.
I mean, one of the arguments has been that the injuries when you fall are not as bad and you repair better. That has been one of the arguments.
Well, it may actually, I mean, yeah, there's a lot of sort of anecdotal evidence that, you know, you see this in the literature that hairy skin heals better. So skin that has active hair growth, because you've got active epithelial cells that are dividing, will actually heal faster or with better quality. I don't know that that's necessarily, you know, been determined, but you know, certainly when you're waxing your hair or depilating the hair, you're continually driving those cells to regrow or regenerate the follicles, right, that normally wouldn't. So they're no longer in their resting state. As soon as you pull out those hair, the hair, you know, they're being activated to regenerate. Not the same as when you shave. So if you just shave, like your leg, they're not regenerating. They'll sit in a state of dormancy. The actual depilation itself causes micro-injury, and then it will activate those cells to always grow. So it's almost like they're primed to heal.
Of course, the laser removal of hair that then supposedly doesn't grow, so I assume then you destroy something completely.
Yeah, in that situation, you're essentially destroying the epithelial stem cell population. So you'll cause micro-injury, and then eventually enough damage that you'll deplete that population in each of these follicles, and then they won't ever grow back again.
So does all skin have hair follicles?
No, so the palms of the hands and feet won't have any hair follicles, but the majority of the rest of the skin has small thallus follicles that are there.
So is the wound healing then in my hand essentially different from the back of my arm, for example?
I mean, it's reasonably good from the palms of the hand. You likely have different cells that are involved in the process, but you know, healing on the palms and the glabrous skin on the feet is actually not so bad. So I don't know if it's there. I mean, it would depend how you measure it in terms of. I don't think there's, you know, if you're looking at severity of scar and things like that. I mean, I don't know.
Ventral skin rises differently from dorsal skin. There may be some defects in things like that.
Yeah.
And there are some people who aren't have no hair on their body at all.
So they have no follicles at all or no hair?
Yeah, they have follicles, but they'll have a mutation and they won't just generate a fiber. Yeah.
Follicles are there now. So you talked a lot about the injury line and how that might be important. So is it correct to assume then, you know, if you have a cycling accident in the mechanical area of your thing compared to a burn, compared to an acid injury, so that would actually then be very different repairs?
Well, I mean, essentially, when it comes to the clinical care that you receive, it's essentially the same thing. And you're going to just, in every one of those situations, it's a full thickness injury, you're going to come into the clinic or into the OR, you're going to debride that whole wound down to the muscle, and you're going to get a sting graft over it. Right? That's the standard of care, so they would all receive the same thing. But I mean, like, yeah, I think the difference is maybe something like in a chronic wound versus an acute wound, where you have differences in the environment there that may change the way the cells respond to that wound. I mean, whether the same thing happens in humans, we don't really know. I mean, but I think that like the take home from this is just simply that that by changing the cues that the cells see within a wound, the same cell can be driven to do two entirely different things. And so the environment seems to be very important. So you know, you've got the same population that's being recruited in, but it sees one set of signals and it becomes fibrotic, it sees a different signal. And so
it's been brought up already a couple of times, the different meaning that you have in the center compared to the periphery. So you have hinted at the idea that maybe in the chemical environment there is different,
yeah.
Different. We have evidence for that.
Well, we have evidence based on isolated cells, and so when you, then even just putting them on different substrates with different properties, it changes the way the cells behave. So one, it maintains them as a stem cell, and they just continually divide, and they don't give rise to any differentiated progeny. And then in the other situation, they entirely commit, and everybody differentiates. So whatever the mechanical properties are, they're driving gene expression programs that are telling them to do two different things. So the same thing may be recapitulated in the womb, right?
You're hinting at the idea that stiffness would be very different in the center compared to the
Yeah, you may have different tension at the periphery than you do in the center. That may be, or you may have a different composition of cells within one area versus another that might change the environment there as well.
Is there any attempt to put a graft down to mechanically stimulate, or I assume you just try to keep it as quiescent as possible and not move it at all?
Yeah, it's basically secured so that it doesn't move. So you're stapling that thing down
So
that
you not go in there mechanically trying to stretch it out in different directions as it is healing to see whether or not that.
No, typically their main concern is they don't want any movement. So any sort of shear on that the graft will fail. So they really want to secure the whole thing with staples so it doesn't move for a period of 10 days, and then they wrap that with bandage so that it maintains it in a single state. So, but I think by changing some of the or applying some substrate beneath the graft to change, because you're still getting fibroblasts that are coming in to fill in that space beneath the graft, right? And so if you could add some sort of substrate that changes the way they behave once they come into the graft, because their natural response is to become fibrotic in a big way, and that's why you see those results like that woman had with her grafts. So if you could provide that either with instructive cues or with a mechanical environment, maybe that's a softer environment that the cells come in and see, and maybe they have a different response and they just make a less fibrotic dermis beneath the graft.
We're talking also about the rat scratching and some animals when they have wounds, they lick it.
Yeah.
And some people have argued that there is something special about the saliva.
Yeah, like dogs, for sure.
Is there anything scientific to that the licking actually will promote healing or so? I know cats have very rough tongues and they will actually eat their skin away by licking continuously.
Yeah. I don't know if there's any sort of antiseptic sort of properties of the saliva. Certainly that you hear that about dogs, right? That this is one of the reasons why they lick wounds is to kill off some of the microbes that may be in the wounds, but I don't know about that.
But you don't think that the licking or the saliva or the mechanics of it would somehow enhance your progenitor cells or anything like that?
Well, in terms of sensory stimulation, I think that may be. So stimulating, because one of the things that we know for sure is that nervous input is critical for regeneration of the tissue. So, like for example, like an axolotl. So these lizards that are able to regenerate limbs, if you cut off the axolotl limb, it will regenerate over a period of several weeks. But if you transect the nerve that innervates that limb a few weeks before that amputation, and then you do the same amputation, it will not regenerate. So there's something about having nervous input within the tissue that's sort of orchestrating these events. So this is one of the other reasons that we want to sort of try to mitigate scars so that we can get better reinnervation of the wound bed in order to enable those instructions to happen.
So in a healed wound, you'll have feeling again in the skin at the end of the day.
Yeah, to some degree. A lot of times they're completely insensate because there's scar or there's something there that prevents innervation again. So like it'll be essentially numb, but then you'll have this chronic itching and sometimes painful sensation in there, or it will be very cold. And so it's certainly an abnormal sensory effect on it, particularly after grafts.
Well, we'll have to look at whether that's ion channel mediated, because that's presumably something that gets your urine further up.
Yeah, I have no idea. I don't know if it's actually. I would imagine it will be to some degree, because in some instances you get reasonably good reinnervation. It's the same with like a sensory nerve injury. You get really good. Like if you have a nice clean transection of your nerve, it will actually regrow, and you get new fibers within the skin and within the musculature. But you still have abnormal sensation, and so I would imagine at sort of the ion channel level that whatever that information is that's coming is being improperly sort of transmitted, and so it would probably be at that level.
I think in the nerve transection, it's very well known that the motor commands essentially get fully reestablished, but the sensory channels for some reason don't connect properly.
Yeah, absolutely.
And I'm not sure why that is, but there's a structural explanation for that.
Yeah, I mean that's one of the things that was in the little bio. So it's, and maybe I always think this is karma, because so we studied peripheral nerve regeneration in the lab, and we used a transection model of the sciatic nerve. And so I guess it was 6 years ago, I was out skiing at Lake Louise and had a bad fall in a turn, and went over my ski and degloved my leg. So from about here up to my knee. But in doing so, I beautifully transected my peroneal nerve. Almost exactly the same thing we do in the laboratory week after week. So, and it's exactly that. My motor function came back after about 15 months of sort of having a dropped foot, but sensory function never came back. So there's a real difference between the capacity for motor fiber regeneration versus sensory fiber regeneration.
Because I know in cat studies, neuromuscular cat studies, they use exactly that model, because the motor comes back into sensory as well.
Last question.
So in one of your early slides, you showed that below the nerve fat. And as we get older, our skin seems to get thinner, and that fat layer disappears. Do you think there's a role for that fat in some sort of like miracle relationship with maintaining the cells in the nervous as well as and contributing in different ways to the integrity of it?
Absolutely. And I think that's one of the reasons why. So we see that the progenitor cells in the hair follicle, they decline big time. As soon as about middle age, they just start to drop off. And you see this in mice and in humans. And so we've done experiments looking at like analyzing these cells over 2 years in a mouse, and you can see them decline a lot. But you're absolutely right. The fat declines at the same time. And one of the things that the fat does is it produces lots of different growth factors, one of them being PDGF. And we know that PDGF is absolutely critical for maintaining the dermal progenitor population. So when we knock out receptors for PDGF signaling, the population declines. It's almost like an accelerated aging phenotype where they just drop off, and then they're not able to regenerate hair follicles as well. So I think that's one example, but I think the fat does so much to maintain not only the stem cell pool, but also the maintenance of the vasculature, those cells within the vasculature, lots of stuff. And I think it's really an underappreciated component of the skin, and probably any other organ as well, too. So, and we're just starting to understand some of the things that it does, but yeah, and so I think the role in aging is really, really important.
We had done some pig studies a while back, and the skin, they built the skin down to the top of the fat layer. We didn't get an abnormal hair. But the fat layer off, then we got contracted scar. So that fat, it disappears after a while, but we don't know what's regular in that fat either. And I guess it does innervate it as well.
Oh yeah, a lot. Yeah, and I think actually maybe that's one of the key things that it does, because of all these growth factors and neurotrophins that it provides. It may be really important for actual reinnervation of skin and maintaining the nerves there too. Because even in the mouse, like over the cycle, over the hair cycle, the fat is increasing and decreasing, so it's constantly being repopulated all the time. So if you lose that ability, then it just will gradually fade away, and then has downstream effects on many different cell types. Yeah.
All right. Let's thank Dr. Biernaskie. Next week we will have Dr. Juan Núñez, PhD student, Danilo Llaneta, sharing the results of his work on the oxygen extraction signal and the limit of exercise tolerance.